Life Sciences from Zurich in 1989, and I guess that's the PhD equivalent. And in Human Medicine from the University of Lausanne, 1994. And again, that's the MD equivalent. So over the course of his career, he has led research in teaching departments, coordinated the Swiss National Center of Competence in Research, TransCure, and served on the presidency of the Swiss National Science Foundation. Since 2022, he has been the Vice Rector for Research and Innovation and a member of the Executive Board of the University of Bern. Since 2014, Professor Abriel has engaged in sustained scientific collaboration with junior and senior colleagues across the African continent, as you might have heard already, initially in the Democratic Republic of Congo and Morocco, and more recently also in Senegal. Cultivating equitable, respectful, and inclusive partnership with African universities has become a central priority for his academic work, reflecting his commitment to fostering a global research environment that is fairer and more accessible to scholars in both the global South and the Western world. His primary research interest concerns channelopathies, rare human diseases caused by ion channel dysfunction, including cystic fibrosis, cardiac arrhythmia, and epilepsy. His group also developed nano-based DNA sequencing protocol to investigate clinically relevant genes involved in rare and monogenic disorders, with a particular focus on African population whose genomic diversity remain underrepresented in global data set. Please join me to welcome Dr. or Professor Hugues Abriel. And thank you very much for making the time to come and visit us here at SUNY Downstate. Thank you.
So, dear students, dear colleagues, dear Ahmed Boujdir, dear Dr. Boujdir, so it's a pleasure to be able to present here. It's going to be about science, but it's going to be also about a journey. And actually, it's a personal journey that started about 10 years ago for me when I first went to Africa the first time as a scientist. And I met with these two people here, Jerry Mumbungu and Claude Kayembe. There are two pediatric doctors that I met in 2014, 2015. So my life changed after that day when I met them. And I thought, well, as a scientist, I could do something different from what I was used to do. I had a classical career as a scientist. I had also my postdoc here in New York, 2 years. Then I went back, and so I climbed the ladders of academic, so assistant professor. But then somehow going to in African universities and meeting with colleagues there has completely changed the way I wanted to proceed and do my science. It's just not only about science, and it's going to be a bit about, this journey that I'm going to mention. Channelopathies. What are channelopathies? So channelopathies are diseases that are caused by ion channels that are not functioning well. And there are many types of channelopathies depending on the organs where these ion channels are located. And I've been doing a lot of research on cardiac channelopathies. And when an ion channels, be sodium channels, calcium channel, potassium channel, doesn't work well, or are blocked or are mutated, you have so-called cardiac arrhythmias, such as here this torsade de pointe. One can classify these different channelopathies. We have the inherited congenital genetic channelopathies and the genes that are mutated here are genes that are coding for the pole-forming subunit, sometimes the associated subunit like beta subunit or CHIPs, we call them, ion channel associated proteins. But we also have, and this is a topic that has been studied by Dr. Boujdir over the years, we call them acquired channelopathies that are caused either by drugs, because those are side effects from drugs, or antibodies, autoantibodies. This is the specific research topic of Dr. Boujdir. Okay, so that's what I done here. I learned to do that here, or to investigate this field when I was a student, when I was a postdoc, and then over the years, you published and you're more or less successful. But then I was quite interested by something that I learned, that somehow one could use ion channels to sequence DNA. So DNA is a molecule, well, a large molecule, obviously. And there has been possibility, and there have been companies developing this idea that one use nanopores, and actually nanopores are also ion channels, to sequence DNA molecules. The usage of nanopores in biomedical research here are displayed here on this slide. You can do, again, here sequencing of DNA. I will mention that in a minute. You can do proteomics. I mean, some people are trying now currently to sequence unfolded proteins. This is something that will come on the market one day. One can do sensing of small molecules, metabolomics. We can deliver drugs. We can also use these molecules to do different kind of controls. So nanopores are now applied or used in many different fields of biomedical sciences. But here I'm talking about these ion channels that are protein nanopores that can be used to sequence DNA. And these proteins are actually bacterial proteins. You have 9 monomers. And then forms this spore. You see it from the side, you see it from the top, and you see here how a DNA molecule can go through this nanopore. And this is here. You can see that, in fact, by unwinding the DNA with a helicase, then the DNA is actually attracted through this nanopore. There is a membrane here. And in fact, you can record an ionic current that is flowing through the nanopore. And depending on the bases that are the bases of the DNA, the ATGC, you can have different currents that are flowing through this nanopore, because in fact, this nanopore is like a resistance or conductance, electrically speaking. And depending on the base, so the amount of current flowing through it is different. And then when you do that several times, you can multiplex this. And this company, Oxford Nanopore Technology, has developed these devices that allow you to sequence DNA. And I was actually extremely interested, fascinated by the technology. And you will see how I have been using it. So the advantage of this Oxford Nanopore technology for sequencing is that, in fact, this is something completely new. Well, now it's not that new anymore. Since about 10 years, we are using this concept of long reads sequencing. You can do better assemblies. You can look at structural variants of DNA. You will see this is portable, and you will see why I found it interesting to have portable devices. It's indeed quite fast. And well, I will not mention this, but in fact, you can not only sequence DNA, but also RNA and look for epigenetic changes to these DNA and RNA molecules, like methylations. So this is the introduction about the technology that I've been following over the years. And then what has happened is that I had the privilege, because when you are a professor, well paid in Switzerland, they give you the opportunity to take a sabbatical. After 6 years, you can say, oh, I have a project, I go somewhere, and the university pays you. I mean, you just reduce your salary of 10%. And I asked myself, so what I'm going to do during 6 months. And then I wrote a project. I had to convince the board of the university that my project was interesting, academically speaking. I could not, ask for, going to an island and to do nothing. I had to do something. So, and these 6 months sabbatical allowed me to bring 2 of my interests that I started to develop over the years. One was to work with colleagues from African universities. And here is one example. So I spent 2 months at the University of Fez in Morocco, and that's where I met Badreddin El Makhzen, who was at that time a resident in medical genetics. And what you see here already is a portable sequencer of nanopore technology. And this is compared to a normal sequencer, capillary sequencer for single sequencing. So I spent 2 months in Fez, and I spent about 4 months in Kinshasa, where I knew about the colleagues I had here. And this is Jerry Mombongo and Cathy Mombongo, who was working here in this lab. And we tested the idea to use this nanopore technology in these 2 universities. So the second interest was this idea to test these ion channels, these nanopore to sequence DNA. And I'd like to show you something here. Right, so I think, yeah, right. So I'm not sure it works here in this, but what you see here is a trace from one single nanopore that of sequencing cells that I brought to Kinshasa. And what you see here is like a single channel current. I mean, for those of you who may know about electrophysiology, you see here that, in fact, when the pore is open, when there is no DNA in this pore, you have a large current, about 200 picoamps. But when DNA flows into the pore, you have here less current. But this current here is extremely noisy. Actually, this is not noise. The current here is the difference of the current that is generated by the different bases that are going through this pore. And in fact, you can interpret this noise here, if you want, and translate it in DNA bases. So I wanted to, yes, I mean, for some reason that's not working. This would have been, you could have seen here this happening online, but also learned when I was studying, and I was not a geneticist. I'm not still a geneticist. I'm more like coming from the physiology and the biophysics. But what I learned is that for some people, genomics is seen not as a priority for Africa. While my friend Shahida Musa, who is a geneticist, professor of medical genetics at UC at Stellenbosch University in South Africa, while just in South Africa there are 4 million of South Africans that are living with rare disease, and most of these rare diseases are monogenic disorders. What she hears very often is that genetics and genomics is too difficult to do in Africa. Africans would not be able to understand. They don't have the skills. They are not interested. And very often it's said, it's too early. You have other priorities, right? I mean, please look at all these infectious diseases. But just to mention that I had to learn also myself to think about the image that we have here or in the Western world about doing research in Africa and doing medical research on the continent. Well, to be just nice, this is extremely patronizing, right? I mean, very often what I learn is that we come and we infer a lot about what these people really need, our colleagues really need. Another aspect that I learned is, in fact, that Africa and African populations are the most genetically diverse region in the world. I mean, this is nicely here depicted with all these different colors here, very less colors in Europe, in Asia. In fact, these colors of dots represent different types of genetic markers that are found. And in fact, the largest variety of the genomes and the variations is found in African populations. In fact, compared to the rest of the world, each African genome harbors about 25% more polymorphism than each of non-African genome. I thought at the beginning, when I learned about that, it was not clear to me where it came from. In fact, it comes from the fact that, Africa is where, the cradle of humanity. That's where 200,000 years ago, the first modern humans started to evolve. And so the human populations on the African continent are much older than the other population that went out of Africa. And this is just about 50 and 60,000 years ago. And they start to move and to migrate to the other places of Earth, of the world. And in fact, there has been here a bit of a bottleneck, and just a fraction of the variation that you have on the continent went out of Africa. And this explains why you have so much variability of variations in the genomes that in the African continent. So this is a fact. Well, this is an observation, but this is a very important information for when you want to do medical genomics and genetics with African populations, because in fact, what we observe is that we don't have the right reference genomes. So, the reference genomes that we have is mainly a Western genome, a bit of Asiatic input, but a lot of what should be known about the genomes of African population is not yet known. There is another problem. The fact that when we do human genetic studies, so there are many ways to do human genetics, but one way has been to perform all these GWAS studies, genome-wide association studies. When you look at the populations that have been used for the genome-wide studies over the years, so the vast majority were of European descent. And you see here that only 2% population here was African, from a population of Africa, right? And this is a more recent study where you see that over the years, the fraction of European descent populations in these GWAS studies has in fact increased because we do more of them, but the fraction of African descent population here remains stable. Actually, this gap is now much larger. So there is a problem here because, in fact, all what we learn is extremely biased toward, in terms of genomics, toward European descent populations. So that's the other type of aspect of the introduction, and now I'll show you what I did really with my colleagues and when I went there. So the goal of this project was, the idea was to use these Oxford Nanopore Technology methodology to sequence genes that are causing genetic disorders, in particular monogenic disorders. And here I worked in Morocco, but also in other countries with two young colleagues. Nada El Makhzen, who is now a PhD, who is finishing her PhD in our lab at the University of Bern, and the brother of Nada, who is his name is Badreddin El Makhzen. Both were born in Fez, Morocco, and he's a medical geneticist. The genes that were studied with them, Nada studied for above all the gene CFTR. I'll tell you about this gene. This is a gene that is mutated in cystic fibrosis. I studied with Nada's help and colleagues' help the HBB gene, that is a gene that is mutated in sickle cell disease. And Badreddin worked on another gene, which is called ATPB7, that is causing another monogenic disorder that is called Wilson disease. Nada just published her first paper of her thesis, and this is what she's going to be defend in about 2 weeks. This is a study that she performed on demonstrating that we can use this nanopore technology to sequence the gene CFTR in patients that are not from the Western population, but in patients from Morocco. The disease is cystic fibrosis. It's a very well-known disorders. Many variants have been found in this gene called CFTR. Many of them are causing cystic fibrosis. Cystic fibrosis is a multisystemic disorder. Patients die if you don't treat them very young. I mean, they die in the infancy, like before age of 10. If you don't treat them, this is what happens, for instance, in African populations. Nowadays, and we'll come back to that, there are new drugs that are extremely efficient to treat the disease, and the patients, the longevity of the patients is more like 44, 45, 50, and we hope that they will be living up to 60, 70, 80 in the future. So the genetics of cystic fibrosis is well known. In fact, this is a gene. This gene is called CFTR. This is an ion channel. This is a chloride ion channel. It's on chromosome 7. It is a large gene. When you have all exon and intron, it's about 250 kilobases, 250,000 bases. It's very long. The protein itself, it's about 1,500 amino acids. And what we know about cystic fibrosis on the African continent, well, first I have to say that this is a disease that is a recessive disease, that it affects 1 out of 2,000 individuals of European descent. In fact, there is a sort of a founder mutation. This is this mutation that we call delta F508, which is the deletion of a phenylalanine in position 508. This is the most common variant. This is the variant that is the best studied. And in fact, in the white populations, 1 out of 20, 25 is a carrier of these variants. And then what we know about cystic fibrosis on the African continent, in fact, that it affects, at least where it had been studied in South Africa, 1 out of 12,000 people. And in North America, in the African American populations, it's 1 out of 14,000 people. There have been studies of CF in different parts of the African continent, mainly in South Africa and in North Africa. And you see here that's a list of different variants. And indeed, I mean, it comes back to my what I just said before. Many of these variants are quite unique. Those are not the variants that we found in the white population. In fact, as you will see, we'll find many more of these variants in the future. When I started to work on this disease, even though I came from the field of cardiac channelopathies, and then when I talked to my colleagues in African universities, doctors, most of them, they told me, well, we know the disease, but we cannot diagnose the disease. And one way to make a diagnostic of this disease is, in fact, quite simple. One has to just measure the concentration of chloride ions in sweat. And this is so-called the sweat test. And I asked myself, well, how can we do so that my colleagues in Fez, but also in Kinshasa, could have a sweat test device? And we tested that with a crowdfunding campaign in Switzerland, and the Swiss people are sometimes quite generous, and they have the money to do that. And we needed about 10,000 Swiss francs to buy one of these devices. But in fact, we reached about 16,000. So at the end, we could buy 2 of them. And this is the device that you can see here. It's portable. It has small electrodes. I don't go into the details, but at the end, you can indeed record the amount of chloride that you have in sweat when you test with a possible patient. In fact, just to be correct, this is not chloride concentration, but this is electrical conductivity that we measure. And then you can infer the amount of chloride ions that you have in the sweat by recording the electrical conductivity. So we had 2 of these devices in the hospitals of our partners in Fez, but in Kinshasa, and now we have a device in Dakar, one in Nairobi, one in Kigali. And we have a network of colleagues in these different countries who are working on this. Nada's work has been focusing on the idea that we can sequence the whole gene using nanopore devices, these portable nanopore devices, by, in fact, targeting the whole gene by amplifying the gene by PCR, classical PCR, but we do it with what we call long-range PCRs. Maybe you have been doing your PCRs 500 bases, 1,000 bases, 1,500 bases. Here we have special polymerases, and these polymerases can give you at least 25,000. We can go up to 50,000. I mean, we are working on special polymerases where you can amplify extremely long fragments of DNA. And the idea was quite simple. Since we knew that the gene is 250 kilobases, we said, well, 10 amplicons of 25 kilobases. It worked more or less. I mean, actually, we could amplify the whole gene with 11 amplicons, and you can see here all these amplicons, and we could sequence, you pool then all these different amplicons on a sequencing cells, and you get the sequences, and you have a pipeline to analyze the different variants that you observe. And here you may see here that one of these variants is a heterozygote variant because you have two colors. And here you see that there is a deletion of 3 bases. And in fact, this is this delta F08 deletions that you see here on this picture. Is not very well visible, but this is the mutation that, again, most of the white people have. So here, one case, this is a patient that had respiratory problems, also pancreatic insufficiency. A CFTR channel is expressed in many different organs, such as the pancreas. And this patient had a chloride concentration of more than 100 millimolar per liter. Actually, this is the conductivity, but we infer to a chloride concentration that is higher than that 80 millimolar, and this is clearly too high. I mean, we can go through the physiology of sweat production, but in fact, chloride is reabsorbed, but in normal situations. But in the case of cystic fibrosis, sodium and chloride goes to the skin. Actually, some people have, a salt of natrium chloride on the skin. They lose a lot of natrium chloride. And when it is that high, I mean, and with specific symptoms, clearly, I mean, it tells you that this patient has cystic fibrosis. And then here in that given patient, Nada, and again, this is because it is a recessive disorder, found 2 different variants. You see it here. This is a variant that a mutation. Those are 2 missense. I mean, 1 missense mutation here that changed the serine in 364 to a proline. But here, this is in fact a stop codon that is generated here. We were able to find all the different variants when we had a positive sweat test, 2 different variants here. This is here, CF causing. And here, those 2 variants were found in this patient. One, it was known to be CF causing for this family number 2, this pedigree here. Here, for this family number 6, we also had 1 known variant. And the other variants here, the serine 364 proline or this phenylalanine 178 serine, are known at what we call VUS. Those are variants of unknown significance. And most of the time, those are variants that were not found in any database. And again, here, those are variants from this Moroccan population that very well illustrates that you find when you do this kind of genetic analysis, many more new variants. And actually, we increase a lot the number of variants that we can observe in that gene. So what we could do also in the lab at the University of Bern is the fact that we can express this variant in very easy kind of cells, HEC293 cells. Most likely, most of you know that you can transfect them, express the different proteins here. In that case, those are these ion channels. And we expressed, in fact, 4 different variants: the wild type channel, the positive control, which is the delta F08 channel, and then these 2 new variants, the serine, proline, and phenylalanine serine here. And what you can observe is, in fact, and this is the positive control, you observe here that for the wild type, you have 3 bands here, both the whole cell lysate, but also at the plasma membrane, because here we did a biotinylation of the plasma membrane proteins. So here you see that for the wild type channel, you have a clear higher migration band, which is the fully glycosylated ion channel. That's the functional channel that you have at the surface of the cell. So this is the normal pattern that we have with a wild type channel. We knew that the deletion 508 actually makes that there is no reduced maturation, no of such fully glycosylated variant, and this doesn't go to the membrane. So this is clearly showing that there is a maturation process, and most likely it has been shown that this is a so-called misfolding of the protein that makes that this protein is degraded more quickly and doesn't go to the cell membrane where it should be, because this is an ion channel. And then we were pleased to see that, in fact, these 2 VUS would have exactly the same, if you want, molecular phenotype. So there is no maturation for the SP and the FS variant, new variant VUS that we found in these 2 patients. Yes. More importantly, the drugs that I was mentioning that are completely revolutionized the way we treat these patients. This is actually a combination of 3 drugs. We call ETI, elacaftor, tizacaftor, ivacaftor. Those 3 drugs combined together rescue these channels. They can go into the pharmacology, but you can see here that when we treat with the drugs, for instance, for the delta F08 variant, without the drug, there is no fully glycosylated band, but here there is a clear glycosylated band. You see it here, right? And then what we observed is that this was also the case for this F1078 serine variant of that patient, but this was not the case for the serine proline variant here. So we could demonstrate that most likely this patient would be a responder to this drug, but this patient would not be one. This is very important, and this is, it goes into what we call, obviously, precision medicine, because you can just treat the patient that has the right variant here in that case. Good. That's what I wanted to mention now about CFTR, what we could do. With nanopore sequencing, I just mentioned here that, again, there is a paper that Badreddin has now published or is online about another gene which for Wilson disease. And now I will change a bit of the topic. This is portable. So there is a possibility, and this is the working hypothesis, that, because it is portable, because it is quote unquote affordable, actually you can buy the device for $5,000. It makes them a needle possibility source for diagnosing monogenic diseases in places where you don't have any lab. I show you here, this portable sequencing device. So those are 2 versions of it. This is the so-called MK1C, that's the MK1D, even smaller. And then just to show you that you can also have a portable PCR machine that you see it's in these hands. So the idea is now to work on a prototype, a possibility to have a fully equipped medical genetic laboratory in suitcases. So these medical genetic laboratories may enable access to genetic for all. We are currently, I'm working on showing that this is possible. I do it mainly with my African partners. This is a long-term project. I don't know when it stops. And it's a bit of an ambitious and high-risk project somehow, but I have the privilege to be able to spend a bit of the money of my lab for this. This is a picture of a prototype of such a portable lab. This is a collaboration with a company in Germany, Expedit Diagnostics. You see the different instruments here, incubator. You see here the PCR. You can see here a small incubator, vortex, and you can measure DNA. You can do gels. You have batteries, and then you have again these different devices, the PCR device and the nanopore sequencing device that you have here. So when I discuss with colleagues from the different genetics departments in Africa, when I met with them, I told them, well, that's something I would like to test. And the person with whom I discussed this, she is a genetic scientist in Dakar, Senegal, and she told me, oh, let's try to do this with a very easy gene, which is the HBB gene that is mutated in patients with sickle cell disease and beta thalassemia. That's what I did. So, it took us about a year to set up the whole lab. And then last year in July, I prepare all the different suitcases, 3 of them. Here that I live in Lausanne, French-speaking Switzerland. The first trip I had to do is to bring all the suitcases from the lab, Bern, to Lausanne. Well, that was easy, 1-hour train. But then, I took all these suitcases with these instruments, with also all the consumables, but also we still need to have you have to have a cool chain, because most of the kits that use biochemical reagents, they have to be cooled at minus 20. Well, we can come back to that, but at least you have to keep them somehow cool so that they can work, as you know. Not completely true, because actually some of these things you can keep them quite long outside of the fridge, and they still work. Maybe less well, but they still work. Okay, that's what I wanted to do. I wanted to fly from Switzerland to Dakar, test all the things in Dakar, and you will see what I did there. And then after the first test in Dakar, I wanted to go to Kinshasa, where I had my friends, so I had to go to do a stopover in Lomé, in Togo. Kinshasa, I would have liked to test the whole thing again, and then go to a place which is called Kindu, which is a far remote place in the center of Congo, not very far from where there is war now, between Rwanda and Congo. This is a region this is a country that I know. I go there on a regular basis since about 10 years. And I tell you, if you want to learn something about Africa, go there. So I went to Dakar. I was extremely well welcomed. That was this Hôpital des Enfants, Children's Hospital of Dakar, Albert Royer. And those are the geneticists and the doctors and the biochemists. This is Professor Rokhaya, Professor Ndudeme, Professor Fatou. She's the biochemist. And there we tested this device. And this device, or this lab, was focusing on, again, sickle cell disease. I'm sure that you heard all about sickle cell disease. It's, again, a recessive disorder. The gene that is mutated is HBB. This is the beta globin of the hemoglobin protein, or tetramer. And there is a mutation that is found in all these patients. Well, there are several, but the one is very well known. That is also called the HBS mutation, or S allele. And what happens is that when there is this mutation, instead of having a glutamate here, you have a valine. And this mutation, GLUT7 to valine, makes that there is a hydrophobic amino acid. And this hydrophobic amino acid, when you have all the beta chains that are mutated, not just half of them. So you have carriers and they are sort of healthy, but when you have all the different mutation beta-globin chain that are mutated, so when there is less oxygen, venous blood, so the red cells they have these very well-known sickle shaped cell, and they are much more fragile, and that's what causes anemia. But it's not only anemia. There are many different other clinical manifestations of sickle cell disease. Well, anemia, that's one thing, but sometimes, it also plugs the different vessels. It makes a so-called vaso-occlusive crisis, and this is something that kids have when they're very young. It's extremely painful, but also young adults. It's because it blocks the whole circulation. It leads to stroke. There are problems with the heart and different, and because then there is the spleen is not working well, the production of antibodies is not working well. They also have a lot of different infections. So there are actually millions of kids, and mainly, and here this is the epidemiology, that are born and live and survive with disease, and most of them are in the sub-Saharan Africa. As you can see it here, that's here the frequency per 100,000 people. That's up to 1,000 here in these different countries. So we're talking here about Senegal, Cameroon, and all these different countries. And you know it that in the African American populations, there is also this allele that is quite frequent. And you see here that where you have more Black populations here on North America, the southern states, you also have a higher prevalence of that disease. You can treat with hydroxyurea. That's a small molecule, but nowadays we can do also allogeneic hematopoietic stem cell transplantation, gene therapy. It works quite well. It's extremely expensive, but it works. What we did in Dakar, we took, actually they gave me, and we worked together, 10 samples from patients that had a very specific form of anemia. They were not sure whether it was sickle cell disease. So we could extract, in fact, 200 microliters of blood DNA. We did PCR amplification of this HBB gene. This is a very short gene. I will show you. It's very easy to do. We purified, and we did overnight sequencing. Actually, you can multiplex. You can put all the different 10 patients, you barcode, and then you get the results after, in fact, 24 hours. So I just show you here, that's the hospital, that's where we were, Children's Hospital in Dakar. And you see here the suitcase. Here you see the PCR machine to amplify, the laptop. You see that even if you are a PI reaching age of 60, you can still pipette. Again, I'm a bit of proud to show this. I just have to have the right glasses. I did most of this thing myself, but you do need help. And this is the team of bioinformatics. So that's how the team of bioinformatics is looking by Dakar, young women, and some of them are extremely well trained, and they were able to help us with the analysis of the different sequences. Good. What we learned, and here I show you just the results of one patient, but we had 10 of them. And here you see the HBB gene is a very easy gene. We have 3 introns, intron 1, 2, and 3. The length of the gene is about 1,000 bases. 1,100 bases. And what you see here is that when we did the sequencing with Nanopore, we had, in fact, just 2 variants that was heterozygotes, because there are 2 colors here. So you have green and red. So it means that you have heterozygote variants. And these variants, one of them was in exon 1, and one of them was in the exon 2. Actually, you have to read this gene from here from right to left. And here, the first variant in exon 1, in fact, this is exactly this mutation, right? That this is HBS allele, where the normal DNA for glutamine here would be GAG, glutamate, GAG. And here the mutation is GTG, GAG, here glutamate here. And then GTG, it will be a valine here. That's the color here. Red is the T. And you see that this patient is just heterozygotes for that. Just maybe, I don't know if some of you have been doing a sort of NGS sequencing here. The read depth is 1,000, right? We have many reads. We are very convinced that, that this is correct. Okay, so one of the variants is the HBS allele in this patient. And the other variant that is found here in the exon 2, you see here it codes for a lysine. This is a K here. In fact, this is, the mutation is a T. This is red, T, AG, TAG, and then you see here TAG, it gives a stop codon. This is a stop codon. So this patient carries the HBS allele, the pathogenic HBS allele, and most of the patients are homozygotes. They have these two alleles. But here, this patient is a bit special. It has an allele that is HBS, and the other one has a stop codon. And in fact, such a stop codon is what you find in patients with so-called beta thalassemia. In fact, this patient, he's heterozygote for HBS and beta zero thalassemia. This is a compound heterozygosity, and it produces severe sickling disease. And actually, this is clinically indistinguishable from HBSS. I mean, homozygote with the two SS. So, and this was done. I flew on Dakar on the Saturday, and we had the results on Wednesday night with this portable lab. So they never had this possibility to have these, I mean, they can do diagnostics, but they never had this possibility to do molecular genetic diagnostics at this level of precision. And we did that for 10 patients, and for the 10 patients, we had what we call diagnostic grade sequences of these patients with genetic anemia. So we're extremely happy with this, and we will continue to work together. What worked well? I mean, the transport worked well. We got these HBB sequences. This was a portable lab. Actually, this lab would not have to be in a lab or in a room itself. We could, and we've done that. I mean, we the first time we tested this lab, I mean, remotely, it was in a hotel room. We did that because we with patient at sickle cell disease in Switzerland. So we started this collaboration with the Dakar team, and then I wanted to continue to, from Dakar to Lomé and Kinshasa, and go to Kindu and go back to Geneva after 6 or 8 weeks. The fact is that when I arrived at the University of Kinshasa, I lost one suitcase. So one of the suitcases, the one with all the different reagents, with the cool chain and cool boxes, stayed for whatever reason at the Dakar airport. So I knew it actually because when you do these things, you have to think a lot, right? You have to prepare well such an experiment. So I put air tags on every suitcase. And when I arrived in Lomé, I knew there was something that was not right. I mean, I had the air tags. I mean, I could locate these two suitcases, but the third one, I knew, well, there would be a problem. So that did not work well, indeed. So I could not test further in Kinshasa, in Kindu. So this concept of a portable lab is working. I mean, this is a prototype, but clearly it is working. I write here, there is a strong interest. When I tell this story, I present this story to colleagues, they all think it's a very interesting and one can implement, apply. I'm still alone. I'm still trying to get funding for that. We'll see what's going to happen. The whole logistical aspect is really a challenge. Most likely one has to do a bit differently. One way to address the challenge of these cool chain and cool boxes is to work. More and more companies now are trying to develop lyophilized enzymes and kits that you can just transport at room temp. So preparation is important. So the vision is still here. So I'll conclude with a few words. I'm convinced that doing medical genetics and genomics on the African continent is really something to do for many, many reasons that you can. But I have just a strong scientific interest for that, and I share that with my colleagues from these African universities and hospitals. We can learn a lot about the disease from this population. I didn't have much time to discuss that aspect, but I hope that you see the way I present is that there are many, many aspects that one has to be extremely careful about. Ethics, equity, inclusivity. So I learned, I didn't know about the concept of white saviorism. Do we say that? White saviorism? So, the white guy comes and thinks that he will solve all problems that these people have, and they tell you, well, no, that's not my problem. My problem is somewhere else. So I have to be extremely humble, and the way it works quite well, or it works well, I think it's that, when you build a relationship or a partnerships over the years, so that you can really see that you have a trustful and a long-standing relationship. That's very important. So yes, the world changed very fast. I work with these African partners. There are many other parts of the world, that deserve also partnerships and attentions. South America, different parts of Asia. And I think there is a good way, as young scientists like me, as a more senior scientist, to do something good on this world. Thank you.
Thank you very much, Dr. Abriel, and I think this is really inspiring for younger junior students who really want to make a difference in certain parts of the world. I think in this case it's Africa. I'm sure there are other areas in Southeast Asia, things like that. Though what I was going to say, every one of you should have that portable lab for more skills and not working in the UCAD. So